like to thank the Sura Foundation for supporting our work and giving us the opportunity to present our results. What I'm going to talk to you today is a collaboration between the Edward Hines VA Hospital and Loyola University Chicago. And we're looking at ethnic differences in glaucoma. So what is glaucoma? It's the second leading cause of blindness and the leading cause of irreversible blindness worldwide. The disease damages the optic nerve, which is the nerve that carries our visual perception from the eyeball to our brain. There are no early symptoms, and there are a number of risk factors, including age, family history, diabetes, and if you are of African American or Hispanic ethnicity, you're at a higher risk. So that risk is shown here. What we're looking at is the prevalence of glaucoma as people age, starting at 40 years old on the bottom up to the 8th decade of life. And you can see that Caucasians are shown in blue, African Americans in red, Hispanics in green. You can see that for all populations, the prevalence of glaucoma increases as we age, but the increase is even more significant for African Americans and Hispanics. So these are results from a visual field test. This is something we'll all undergo as we get older and work with our ophthalmologists. What we're looking at is what the eye is able to see, and the normal eye is shown in panel A, and light gray means that the eye is functioning properly and you can see, and dark gray or black means that the eye is losing visual perception. So you can see in the normal eye, and then the cross that shows where your central vision is, where it overlaps. And you can see in panel A, that's a healthy eye. We all have a blind spot, but since we have two eyes, it doesn't affect our vision. So that is a normal eye. And as glaucoma progresses, people start to lose their peripheral vision, which is shown in panel B. You can see there's some darker gray and black areas coming on the outside. And then as it worsens even more, you get complete lack of vision, and then central vision starts to be affected as shown in panel C. And then in panel D, you can see this individual has lost almost all sight and just has a little bit of central vision remaining. And since we all like to see, it's important that we try to understand this disease and either prevent it or find new treatments for people that have already developed the disease. So just one more picture of what's happening. What we're looking at here is the optical disc, and the technology is called optical coherence tomography. It's another thing that we're all going to have done at the doctor's office as we age. The optical disc is shown on the top here. This is where all the nerves from the eye that are picking up those visual cues come together, and then the optic nerve goes out of the eyeball, the back of the eyeball, and goes to the brain. On the left, you can see our normal eye. What this depression is called the cup, and that's normally seen in all individuals at the bottom of the optical disc. But you can see in the glaucoma eye that the damage is much more extensive, and there's a real eroding and death of the nerves that are carrying our visual perception signals to the brain. So our approach for this project to try to understand what's happening in this disease is to understand the genes that are being expressed and are changing during the pathogenesis of the disease. So these technologies that do this type of research have been around for a couple of decades. The normal way that people do it is shown on the right-hand side, which I call gene 2, where we're looking at what a cell looks like in the healthy state on the left, and then what it looks like in the glaucomatous state. And traditionally, what researchers are looking for are genes that are being upregulated as a disease progresses. And a few years ago, I started working with biostatistical collaborators to kind of ask a slightly different question. And that's what we call the barcode approach. And in that approach, what we're looking for genes that are not expressed in the healthy tissue and then are turned on during the disease process. So it's a slightly different biological question as depicted here on the left for gene 1. So you can see we're looking for things that cross this threshold from going from unexpressed to expressed. And so we've already got some preliminary results using these techniques. So a number of papers have been published looking at some of this genetics in the disease progression, and they found 15 genes that are changing during glaucoma using the classic approach. Using our new approach, we've more than doubled the number of genes that we think are involved in progression of glaucoma, so to 34 genes. So we think it's pretty exciting. We look forward to sharing more results as the project progresses. So we have a great interdisciplinary team. These are the two basic sciences professionals. It includes myself. I have 10 years of genomics experience. I co-developed the barcode technology, and I'm also in the Department of Ophthalmology at Loyola. And then Dr. Stubbs has decades of experience in glaucoma research, and he works at the VA and is professor and vice chair of research in the Department of Ophthalmology as well. And then we have two clinical collaborators, Dr. Chaku, she's the director of the glaucoma service at the VA hospital, and then Dr. Bouchard is our chair. He's the director of the cornea service, and they're both going to be collecting surgical specimens that we're going to be using in our research. So thank you for your attention.